Hello, everyone. So today we're going to talk about carbon monoxide, and then after that, we're going to get into some analytical overview of the different types of stuff that you've been doing in labs. So sample prep, different types of screening procedures, confirmation procedures, stuff like that. But first, we're going to talk about carbon monoxide. Carbon monoxide is, as most of you know, an odorless, tasteless, non-irritating gas. It comes from the incomplete combustion of carbon-containing fuels, so natural gases, gasoline, stuff like that. It can also come from the partial oxidation of hydrocarbon gases in commercial production. Um, the fact that it's odorless, tasteless, and non-irritating really leads to a lot of the factors that are attributed to its exposure and then therefore its toxicity because you can be exposed to kind of high levels before you even realize what's going on. They have carbon monoxide detectors that I'm sure you guys probably have in your homes. And if you've ever set one off, they're super loud. They sound like a fire alarm, but I think they're even louder. And that is able to detect the carbon monoxide. And you can get different detectors that are either more or less sensitive. Uh, I keep a carbon monoxide detector next to my stove because I have a gas stove. So if something happens and the flame goes out, the carbon monoxide could come from the combustion of the gasoline in the lines. So I leave one there. We have one, I'm trying to think, in our basement, just in case, it's where we have our pipes, stuff like that. And then we just have one in our hallway. So it's really important that you have carbon monoxide detectors all over, especially in areas, like I said, like for me by my gas stove, so that if there is carbon monoxide detected, you can catch it before it reaches high levels and you'll have the greatest chance of not being affected negatively by the toxic carbon monoxide. Some sources. Commercially, it's produced on an industrial scale by the partial oxidation of hydrocarbon gases from natural gas or by the gasification of coal, which is the majority of what is used immediately downstream for chemical synthesis, producing small quantifiable volumes. There's also laboratory production of carbon monoxide, and this is accomplished by heating calcium carbonate with zinc dust or from the dehydration of something like formic acid. The largest contribution, however, comes from highway motor vehicles, and this is a byproduct of highway vehicle exhaust and of household appliances such as gas, oil, kerosene, wood, or fires. So you can see, I don't know if you can see my mouse, but you can see that the cars are what's circled here, and that's because, like I said, that is the leading cause of carbon monoxide. Carbon monoxide exceeds all other atmospheric pollutions combined with the exception of carbon dioxide. But you can see here, especially if you're thinking about like a volcanic eruption or a wildfire, right? It's really easy for these to become very quickly very large scale sources of carbon monoxide. And then as we move into the winter months, right? I didn't talk about it explicitly here, but things like generators, which is actually this yellow bus looking thing, is a generator. So a lot of generators will actually have warnings on them about not using them indoors or using them in highly ventilated areas. And this is because the gasoline that the generators use to create electricity also releases this carbon monoxide as a byproduct of that combustion. So, for the mechanism of action, carbon monoxide gas diffuses rapidly across the pulmonary capillary membrane where it binds with hemoglobin, which is the oxygen carrying molecule in our red blood cells. However, it competes with oxygen, and I use that term very loosely because carbon monoxide has an affinity 200 times greater than that of oxygen with hemoglobin. And when carbon monoxide and hemoglobin bind together, they create a compound known as carboxyhemoglobin. Carbon monoxide decreases the oxygen carrying capacity and then the oxygen delivery to the tissues, which is the cause of cellular hypoxia and eventually ketoacidosis. Our brain is going to be the main target organ for this type of toxicity because the brain has a really, really high metabolic rate and as a result has a really, really large oxygen requirement. So when you're being exposed to the carbon monoxide, right, your brain, which is responsible for all of your homeostatic interactions, right, trying to make sure that your heart rate's consistent, you're breathing, you're thinking, you're doing this, you're doing that, it's going right to your brain. So that's why that's the main target. In the brain, carbon dioxide will bind to cytochrome C oxidase, which results in a decreased mitochondrial function and a decrease in ATP synthase as well. Um, this directly inhibits the ability to do aerobic respiration, and um, it also, in addition to impairing the mitochondrial function and the ATP synthase, it also causes cellular damage and increases levels of peroxidase, proteases, and other reactive oxygen species that are subsequently released from trying to use carbon monoxide or the presence of carbon monoxide in these mitochondria. And then those reactive oxygen species go on to increase cellular damage. Specifically in endothelial cells, proteases form something called xanthine oxidase, which blocks inherent protective mechanisms against oxidative stress. So your body always creates some sort of oxidation stress, right? In your mitochondria, specifically in the ATP synthase, we tend to release reactive oxygen species or oxidative stress that causes inherent damage to the cells. But because of this, our cells also have a way of protecting themselves against this type of damage. And what happens in the case of something like carbon monoxide toxicity is it overwhelms these innately protective mechanisms that we have. So, another example of oxidative stress induced by carbon monoxide poisoning is lipid peroxidation, and this causes the degradation of unsaturated fatty acids and also alters the structure of myelin. Myelin is the substance that covers our neuronal cells, and it helps with the signal transduction pathways. So, it's what essentially facilitates the chemicals in your brain going down those long axons. It's insulated by myelin. And when the myelin breaks apart, it can cause a immunological response and actually cause demyelination. So that myelin sheath that's supposed to be there to help with sending the signals, and it's also supposed to be protective, it goes away. And this causes problems with the central nervous system. And it can cause problems like encephalopathy, migraines, right? It can cause visual disturbances depending on what part of the brain is having the attack from the demyelination. The toxicity is also multifactorial, meaning that it's going to result in a combination of tissue hypoxia with direct carbon monoxide mediated damage at the cellular level. And then it's compounded with the subsequent harm due to oxidative stress, which causes the activation of inflammatory cascades and lipid peroxidation, which then proceeds to release further damaging reactive oxygen species and oxidative stress. Another organ that's going to be highly affected by carbon monoxide poisoning is the heart, because like the brain, it is really, really rich in oxygen requirement, and it's going to be more susceptible to an ischemic injury, which is an inadequate blood supply. So affects like stuff like a heart attack, right? Or even an ischemic stroke, which would be more in the heart. But pretty much anything ischemia is going to be anything that's blocking the oxygen from getting to the cells where it needs to be. The deleterious effects on the heart in particular can vary widely as the cardiovascular health of the exposed person really has to deal with the influences on the compensatory mechanisms. So if somebody is already suffering from heart disease or if they're a heavy smoker, stuff like that is going to impact their health status, as we've learned throughout this course, a couple of times is going to interact with their ability to kind of handle this stress. In hypoxemia, cardiac output increases to maintain oxygen delivery to the brain. So the heart is going to begin working harder when it's exposed to carbon monoxide. And eventually, with sustained and progressive hypoxemia, cardiac output is going to be maximized until it can't compensate for the reduced oxygen carrying capacity in the carboxyhemoglobin. And this is going to make something called carboxyhemoglobinemia. And pretty much what happens in this is there's, again, this ischemic consequence of the cells not having enough oxygen. And this leads to a subsequent reduction in cardiac output, which then further exacerbates the cardiac ischemia because now it's not even pumping this carboxyhemoglobin throughout the body. It's just simply starting to no longer produce, no longer work, and no longer spread oxygenated or deoxygenated blood throughout the system. This is obviously a very vicious cycle, and it's going to quickly lead to loss of consciousness, possibly decreased cardiac output, so the heart stopping, or something like a cerebral ischemia, right, which is when blood flow stops going to the brain. And this has all been identified as a strong indicator of severe toxicity and the ability of the individual to compensate for decreased oxygen carrying capacity is determined by the degree of insult. So, again, like I just said on the last slide, people with underlying respiratory, cardiac, or vascular disease are going to develop severe manifestations of toxicity at lower concentrations than people who are healthier, right? Or maybe people who have a better tolerance of an anaerobic environment. So people who like work out really hard, right, and usually stay in that anaerobic type of respiration throughout their workouts longer will be able to handle this and be able to have less severe consequences at higher levels. So here's a really good chart that shows carbon dioxide levels and their correlated symptoms. So this also tells you that the parts per million that somebody will be able to survive with the effect. So at about 35 parts per million, people will be fine for about 6 to 8 hours. They may have some headache and dizziness with constant exposure, but usually, if you're in this even for a prolonged time, such as 6 to 8 hours, you'll you'll be fine. You'll still be alive. You won't have any of that ischemic injury that I was just talking about. At 50 parts per million for about 8 hours, this is our permissible exposure limit. And our permissible exposure limits are a limit that's set by OSHA, which is the exposure level that a worker can endure for an 8 hour period without having severe side effects. So if somebody's working at, I don't know, a coal processing plant or power plant, something that's going to be creating a level of carbon monoxide, it's legally acceptable for them to have up to 50 parts per million and be exposed to that for 8 hours. So that's the level at which OSHA has determined that carbon monoxide exposure is still going to be safe and not have any lasting effects. At 100 parts per million, if you stay in that environment for 2 to 3 hours, you'll begin having a slight headache, which is kind of similar, right, with the other limits that we've already talked about. However, this is going to happen in a shorter period of time now. If we double that and we get to 200 parts per million, same thing, 2 to 3 hours, it'll have slight mild frontal headache, discomfort, the beginning of loss of judgment, loss of vision, and irritability. As it states here, people should not be exposed to this level. This is already too high and too long to be exposed to a carbon dioxide environment. At 400 parts per million, so again, we're doubling, we will only be able to be in this for about 1 to 2 hours. And during that time, the frontal headaches will continue and people will have the onset of nausea. After 3 hours at 400 parts per million, the status of the person will become life-threatening. If we double again to 800 parts per million, people will only survive, people will only really be conscious and like able to survive for about 45 minutes. They'll have dizziness, nausea, convulsions, and collapse within about 2 hours and possible death. At 1600 parts per million, people only have about 20 minutes before they start feeling headache, tachycardia, so their heart racing, dizziness, and nausea. This will also be compounded with other central nervous system factors such as confusion and staggering or ataxia, loss of gait. And death is going to occur in less than 2 hours if you're constantly exposed to this level. Now we're getting into the really high numbers. So at 3,200 parts per million, 3,200 parts per million, people will only have about 5 to 10 minutes before they start experiencing headache, dizziness, nausea, and then they will be unconscious in about 10 to 15 minutes, with death about 20 minutes after that, 15 to 20 minutes after that. So at about 30 minutes. So the reason that there's also kind of this discrepancy between the time of death and the amount of time that people should be exposed is because that time column is telling us the time at which the person will start feeling the effects that are listed in the right column, just in case that was confusing for anyone. At 6,400 parts per million, people will start feeling the effects within 1 to 2 minutes: headache, dizziness, nausea, convulsion, respiratory arrest, and death within less than 20 minutes. And then finally, at 12,800 parts per million, people will only have about 2 minutes that they're really able to be in there. They'll be unconscious within 2 to 3 breaths because of the amount of carbon dioxide that's going to be competing for oxygen and turning into carboxyhemoglobin, with death occurring in about 3 minutes. So after about 3 minutes in this high of an environment, you'll only be able to take about 2 to 3 breaths. That'll completely deplete your oxygen levels. And then at that point, death will occur. So people who are smokers are able to tolerate higher levels of carbon monoxide because they actually kind of have like a steady state level of carbon monoxide already in their system. So they're already functioning at a lower oxygen saturation than the rest of the general population due to the carbon monoxide that's they get when they're smoking the cigarettes. This also brings up another interesting point. So cyanide, so carbon monoxide and hydrogen cyanide are common proteolytic products of plastic materials and fires, and they actually interact synergistically. So if you remember from one of your other talks classes, right, that we can have additive effects. Dr. Ship might have gone over it, but you can have additive effects and you can have synergistic effects. And so an additive effect is pretty much saying if you have 1 plus 1 equals 2, whereas a synergistic effect will say if I have 1 plus 3 equals 25. So instead of them compounding each other in an additive fashion, they'll multiply exponentially. So you'll have a much greater effect if you're exposed to carbon monoxide with the hydrogen cyanide. Um, Hydrogen cyanide is going to inhibit cytochrome C oxidase, and this is again going to prevent aerobic respiration by inhibiting the terminal step in the electron transport chain of the cells of the brain. And this is going to result in severe lactic acidosis. So again, this kind of relates back to our biochemistry, right? Where we have the cytochrome C oxidase, the electron transport chain, all of that. So this is happening in the mitochondria, which are responsible for making most of the ATP that we use, right? And pretty much what's happening here is the last step of cellular respiration results in a really high output and of energy. And when we inhibit that part of the electron transport chain, we're stopping that energy from being made. And then again, like we were talking about earlier, we're still increasing the amount of reactive oxygen species and oxidative stress that the cells are being exposed to. Cyanide causes rapid onset of central nervous system and cardiovascular dysfunction, which results in a loss of consciousness, respiratory distress, or respiratory arrest, and death. So again, this is going to compound all those numbers that I read before. If there's the presence of cyanide, it's going to be happening much, much faster than if somebody was just exposed to the levels of carbon monoxide. So thankfully, there is some treatment. There's oxygen therapy, supplemental oxygen, ventilation, and hyperbaric chambers. However, to add insult to injury, reoxygenation and reperfusion following periods of hypoxia generate partially reduced oxygen species, which can then further oxidize proteins, nucleic acids, and cause further inflammation, sometimes even leading to apoptosis or cell death. And this is pretty much analogous to a reperfusion injury, which is described in other acute ischemic processes. So sometimes when we have an area that is ischemic, meaning that it doesn't have proper blood flow, and we reoxygenate the area, the reactive oxygen species that are generated from oxidative stress actually lead to further cellular damage than just the ischemic injury. So it's kind of a double-edged sword. So obviously, how does this relate to things like forensic science? So fire deaths. With fire deaths, we have carboxyhemoglobin levels that allow postmortem examiners to determine if the person died before or after a fire was started. Carbon monoxide poisoning can affect cognitive ability, causing escapability to be a problem in a fire setting. And then that compounded with things like drug intoxication can reduce the ability for someone to escape. There's also some indicators about fire deaths, right? You can see differences in the red blood cells or in the person's blood, right? The color of it could be different if it's oxygenated blood versus deoxygenated blood. So if it's deoxygenated because of the binding of carboxyhemoglobin, it's going to be much brighter than that kind of rusty dark color that you normally see in oxygenated blood. So it could also be another indicator, the color of the person's blood for fire death. It's also possible, right? We talk about that a lot of the times this will happen if somebody's asleep because their brain isn't even awake enough, or obviously they're not awake if they're sleeping, but they won't be able to kind of see that they're having like the headache or maybe they're having problems with their vision. And it's just very quickly going to result in unconsciousness, which is then obviously also going to reduce the escapability of the fire situation. So that's carbon monoxide. We're now going to switch gears a little bit and we're going to talk about analytical analysis. So this is an analytical overview. We're going to start with different types of samples that you might encounter. So some common clinical samples are urine, peripheral venous blood, which is like the blood that's taken out of your arm during a blood test, oral fluid, and hair. And then some forensic samples we have, again, urine, peripheral blood, usually femoral blood, vitreous humor, which is located in the eye, tissue samples, so this might be liver samples, brain samples, and then we also have hair, which is not a postmortem sample. Vitreous humor tissue samples tend to be postmortem. You're not really going to be testing those for drugs when people are alive. We would use an alternative matrix like urine or blood. Additionally, we want to take femoral blood because we want to not, if there's any sort of injury, we don't want to do blood that might be tainted with something else that's happened. Or we also refer to that as postmortem redistribution. So blood contains obviously the blood itself. which we think of like the red blood cells, the white blood cells, kind of the other components, and then also plasma and serum. This allows us to have a window of detection of a few hours to a few days, depending on what the drug is that we are testing for. But most importantly, blood allows us to correlate a blood concentration with drug effects. Another good thing about blood is that it's not easily adulterated, right? People are taking it right from your vein. There's not much you can do to mess it up, unless maybe you're knocking the vial of blood out of someone's hand. But um, there's really no good way for patients to adulterate the sample. Some downsides is that it is an invasive collection. Obviously, somebody's getting stuck with a needle. You need trained medical personnel to do it, right? So most cops aren't going to be able to do this. You have to take the person to a hospital where they can have a blood drawn by a phlebotomist. And it's also a biohazard. Blood is a biohazard. So there's special precautions that have to be taken along with that. Next, we have urine. The window of detection for urine is, sorry about that. The window of detection of urine is hours to days. There's a delay between drug consumption and the drug being in the urine. So immediate exposure is probably not going to be detected. Uh, with urine, because it's stored in the bladder for an unknown amount of time, right? Usually several hours. We're not able to correlate a urine concentration with the drug effect. It pretty much just tells us, did this person take this drug, yes or no? Drug present in urine does not mean that there is drug present in the blood. So again, because of this delay, it's definitely possible that by the time a urine drug test is done, there's no longer that drug in the blood and it's not having a direct impact on that person's behavior. Um, a upside to using urine is that we have large amounts of it, right? Somebody's going to be able to release a lot of urine and it's going to be usually in a high concentration, especially if it's urine that's been accumulating over several hours. There are some problems with it. It's very easily adulterated, diluted, or substituted. However, we can do things like test creatine levels and specific gravity to test for any sort of adulteration or dilution. And usually we don't find the presence of a prodrug in urine samples because it's already been metabolized. It's definitely possible that some parent drug is excreted in the urine, but prodrug is not really going to be found. Next, we have oral fluid. Oral fluid contains saliva, gingival, cervicular fluid, and cellular debris. Oral fluid is a filtrate of blood plasma, meaning that we actually are able to correlate concentration of drugs in oral fluid. So this can kind of give us a snapshot into how high the concentration of the drug is in a person's system at the time the oral fluid is taken, and then could be used to calculate um, the levels of the drug in the person's system at the time of whatever happened. Drugs must enter the oral fluid through passive diffusion, so it is kind of limiting because this is mainly going to be molecules that are small, unbound, non-ionizable, and lipid soluble because they're going to have to diffuse through the plasma membranes in the inside of the person's mouth. This is also a good matrix if you're worried about things like adulteration. Uh, Saliva is remade, so every couple of minutes, your body just naturally swallows your saliva. So if you have somebody sit in a room where they don't have water, they don't have access to anything that could be used as an adulterant, after about 15 minutes, they're going to have clean oral fluid. So even if they like drink something beforehand, if they sit and wait, eventually that oral fluid is going to be remade and then it's going to be collected. And what's nice about oral fluid collection is that it's non-invasive. There's the Quanticell. I had a picture of it on the other slide. I don't know if I go back. Anyway, the cell collection devices, you just put them underneath your tongue, and there's a little indicator on the end of it. And after however long when I had to do this in the lab, I think it would be like 5 minutes at the most. The collection device, the light changes color, not the light, the sensor changes color, and you know that you've collected enough oral fluid for any sort of toxicology testing. Next, we have hair. Hair is a really unique matrix because it gives us a really long time frame into exposure. So it can be weeks to months. So what happens with our hair exposure is, or our hair testing is hair grows from the scalp, and in the scalp, it's nourished with blood flow. When this hair is being grown, it will also sometimes absorb the drugs that are circulating throughout the blood system, and they will bind to things like the melatonin or sulfhydryl containing amino acids in the hair, like cysteines. And um, this allows us to test hair. As you can imagine, right, because it's just the single hair getting nourished from the blood flow, the levels of drug in hair are really, really low. So we usually measure them in a different unit than we would other things like drugs in urine. Our hair is primarily protein. It's about 65 to 95% keratin, which is the same thing that our nails are made out of. And we combine that with our hair being about 15 to 35% water, and then about 1 to 9% lipids, and then about zero point 0.5, 0.25 to about 0.95% minerals. So our hair doesn't actually grow in cycles. Sorry, our hair doesn't continually grow, it grows in cycles. So this can also be something that has to do with like hair length, and at what point the drug is deposited into the hair. So the first phase of hair growth is the anagen phase, and at this point, the follicle is actively producing hair, and this takes about 4 to 8 years for head hair and about less than a year for non-head hair, with a growth rate of about zero point 0.6 to 1.42 centimeters per month, depending on the hair type and anatomical location. So usually we're talking about hair that's coming from the head, but there's also pubic hair that could be tested, body hair, leg hair, arm hair, stuff like that. Next, we go into the catagen phase, and this lasts about 2 weeks. In the catagen phase, cellular division stops and the follicle begins to degenerate. Then we have the telogen phase. So at this point in the catagen phase, once that follicle begins to deteriorate, there will no longer be a blood supply. So at that point, the hair is not going to be getting any longer, but there's also not going to be adding any drug into that hair. Then we have the telogen phase. This is a resting period for our hair. It lasts about 10 weeks. And at this point, the hair shaft is going to completely stop growing. And then the hair naturally sheds and falls out. So, some problems with hair testing is that um, there's a lot of factors that impact hair growth. So, age, race, disease status, nutritional deficiencies, and pregnancy are all aspects that can be known to influence the rate of hair growth or the length of the hair in the rest period before it falls out. There's also cosmetic treatments that may alter drug concentrations in the hair. So things like people bleaching their hair are going to, if you're bleaching your hair, right, you're removing the color from it. So you're removing those melanin molecules that are in there, and that's going to release some of the drug that was bound in the hair. And then this also, because drugs bind to melanin, which is the colored pigment in hair, this could also lead to a racial bias, such that people who have darker hair are more likely to have a greater concentration of drugs in their system, and that could lead to some sort of a bias. Next, we have vitreous humor. So our vitreous humor, this is going to be a postmortem sample, and this is a clear gelatinous substance that fills the eyeball between the retina and the lens. It is an isotonic salt solution with very little protein content, and it's usually similar to post-mortem blood. or the time, right? It's usually similar to the blood concentration at the around the time of death. Collection involves a needle being placed into the eye, and then the target compound is going to be either drugs or their metabolites. So just like things that you would find in peripheral blood. And this can determine recent drug use. Its window of detection is also going to be hours to days or even less. Again, it's going to be very similar to blood concentrations. And we can relate pharmacological effects of drugs to vitreous humor since it is similar to our blood levels. Finally, we have tissues. So tissues are going to be collected for postmortem samples. So these are also going to be more forensic samples. And tissues are liver, kidney, skeletal muscle, brain, lung, heart, and bones. The problem with tissues is that it's very difficult to interpret because tissue concentrations do not correlate with blood concentrations. And sometimes there are going to be a few different data sets available. It's really just more of an indicator of recent drug exposure, hours to days, or past exposure in things such as bones, right? Because your bones take a really long time to remodel. So that'll kind of give a longer snapshot. But for the most part, your tissues are just going to kind of hold on to any sort of drug that was deposited from the blood around the time of death. So those are different samples that we could work with as toxicologists. Next, we're going to move on to sample pretreatment and extraction. So sample pretreatment or sample preparation in general, this includes some aspects of extraction, are going to be the removal of insoluble residues and interfering compounds, the concentration or dilution of the analyte, the disruption of protein binding, protein precipitation, hydrolysis of conjugates, homogenization of solid samples, the addition of internal standards, and the turbidization of analytes. So for sample pretreatment, it's going to depend on the type of matrix that we're using. For blood, we are mainly going to be doing protein precipitation, and we are going to be doing this using an acid. So before the extraction procedure can begin, it's necessary to determine the total drug in the plasma of the blood and to remove interferences for the extraction analysis. So some sample examples of things that we're going to be removing are going to be things like our proteins that are in the blood, any type of white blood cell, stuff like that. So we really want to concentrate the drug into the watery layer of the blood. Some solvents that we're going to use for protein precipitation are going to be acids or organic solvents, because these are going to make those proteins kind of crash out, what we call crash out in the solution. And this way we'll be able to easily separate them. For urine, we may have to do hydrolysis, and this is going to be required to get the free drug that's going to be analyzed using certain techniques, because some things such as gas chromatography can analyze conjugates in the blood. So we have to hydrolyze them so that we're able to then analyze them. Next, we have oral fluid. So for this, we are going to usually correct the pH. So in order to properly extract oral fluid, you want the pH of the solution to be either two full pH values above or below the pKa of the drug of interest to ensure the best possible interaction between the analyte and the sorbent pad of whatever SPE you're using. This is done depending on the type of extraction that will be determined later on. So you'll be able to determine if you want your analytes to be ionized or unionized. If you're looking for an acidic substance, an acidic drug, nearly total ionization is going to be observed at two full pH units or more above the pKa value. And then on the other hand, no ionization or very, very little ionization is going to be observed two full units below the pKa value. In comparison, for basic substances, nearly total ionization is observed at two full pH units or more below the pKa value, and no ionization is going to be observed two full units above the pKa value. For hair, we have several steps that are involved in our pre-treatment. First, we decontaminate the hair to remove any external contamination. This is really big and really important for things like somebody who, right, might be getting tested for THC, but if they're around somebody who's smoking marijuana, they will be, they'll have it on the outside of their hair. So the decontamination process removes that external contamination. Next, we segment the hair. So essentially, we do this to make a calendar. So we're going to cut the hair at um, certain lengths to determine how many months of time we're going to be looking at. This is again a little bit of a problem with hair because not everyone's hair is going to grow the same way, the same amount of time, the same length. Um, so the segmentation of hair is going to help us to determine that calendar. Next, we do homogenization, which is when we cut the hair into really, really, really small pieces, or we essentially pulverize it. You then weigh out a certain amount of the homogenized hair, and then digestion is done to extract the drugs from the hair. Next, tissue. So tissue samples are also going to be cut into really, really small pieces, so that it can be properly analyzed. So first, you're going to cut them into small pieces and weigh them so that you're able to make sure that you're consistently using the same amount of whatever matrix, whatever tissue it may be. And then they're going to be further homogenized in a buffer using something like a blender or a pulverizer. And then later, they're going to be extracted or homogenized in the buffer again. The obtained liquid from the homogenization is what's actually going to be extracted through whatever extraction process is used. Now we're going to talk about extractions. So extractions is pretty much just the separating of a target substance from interferences within the sample. The fundamental principles, there's a lot of them, but some of them that I wanted to highlight are solubility, distribution equilibrium, state of ionization, and then the introduction of the internal standard. So the solubility is the ability of a substance to dissolve in a particular solvent. The solubility of a substance depends on the chemical and physical properties of both solute and solvent. So you'll hear us a lot of the time when we're talking about extractions in lab or something like that, talking about if the drug we're looking for is polar or nonpolar, and that helping for us to determine what type of solvents we're going to use. So right, for something like THC is very, very lipophilic. So we're not going to use like an aqueous solution or something like that to try and extract it because it doesn't want to go in the watery solution. It would rather go in an organic solution. We will also use the distribution equilibrium. And so this is the concept that the distribution of ionized substances or non-ionized substances between the aqueous phase and the organic phase of the solvent. So again, this is going to correlate to what type of extraction we're going to end up doing, right? If we're using reverse phase or normal phase or cations and stuff like that, it's going to help us to determine where in the extraction process we'll actually be able to track the drug. Next, we have the state of ionization, which I already talked about when I was talking about oral fluid, right? And so this is according to the Henderson-Hasselbalch equation. Acidic or basic substances are 50% ionized at their pH value, equal to the substance's pKa value. So that's why we want to adjust our pHs either to +2 units above or +2 units below the pKa value to make sure that we have 100% ionization or unionization of the drug, depending on what type of extraction procedure we're doing. And then finally, we have the introduction of the internal standard. The internal standard is going to be a deuterated analyte. So it's going to have some sort of molecule that is adding weight to the drug. So it's going to essentially be a drug molecule with an added hydrogen or something like that on it that's going to change the weight. And this method of analysis allows us to compensate for, or at least address potential sources of error. Some potential sources of error could be the volume or weight measurements being incorrect, losses on the surface of containers, or because of evaporation, the addition or transfer of steps, or also instrument problems like problems with injection, transfer to the column, or detector drift. They also aid us in analyte identification. So with an internal standard, we're going to know its relative retention time, as well as its precursor and product ions. So we're going to know exactly what we want to look for. And then we're going to be able to use that internal standard as kind of like a control group that we use in every single sample that we're analyzing, which is why, right, when you're doing your labs, you're still going to put the internal standard in the blank because you want to see if something's happening along the way with any of the samples. So it acts as an internal control that we can use to make sure everything's going right in our extraction process, as well as something we can then use to help with analyte identification and quantification later on. So there's a bunch of different type of extraction procedures. I just pulled a couple of them that I wanted to highlight that are ones that we either do often in the labs here or are of particular interest in a clinical or a forensic lab setting. So one of the first ones that we're going to talk about is a liquid-liquid extraction. And this is extraction of a target analyte from an aqueous solution to an organic solvent by mixing the two solvents until an equilibrium is reached. And at that point, the drug will be hopefully wherever we want it to be, right? So again, using THC as an example, we're going to get our THC into our organic solution because that's where it's going to be most happy. Some advantages of liquid-liquid is that it is fast and inexpensive and efficient. Our extraction yield is going to be determined by the solubility and distribution equilibrium of the analyte in the organic solvent, as well as the ionization status of the analyte, the equilibrium of the electrolytic dissociation. So that just means that it's going to be pH dependent. And then we're also going to have to take into account the ratio of phase volume, so the ratio of our organic to our aqueous solutions. And then we're also going to have to take into account the number of extraction phases, stages that we do. So usually in our labs that we're doing in either the grad program or in Tox 415, they're going to just be simple liquid-liquid extractions, but sometimes you might have to do either back or double back extractions. And this is stuff that you might have encountered in instrumental. Sometimes you'll have to do back extractions with your general unknowns, and pretty much that's just you're putting the analyte of interest into either the organic, and then maybe you're moving it into an aqueous, and then you want to move it into a different organic. And pretty much what the doing multiple liquid-liquid extractions does is it helps to kind of purify your final product so that you're going to be able to get a better result. Next, we have our supported liquid extraction. Supported liquid extraction consists of the immobilization of the aqueous phase, such as diatomaceous earth, inside a cartridge prior to extraction. The organic solvent is going to elute the compounds of interest, and this eliminates the emulsion formation that can increase the extraction yield of certain analytes. An emulsion is something that can happen in a liquid-liquid extraction where the organic and the aqueous solution kind of mix together. And this makes it really, really difficult to separate them out because you'll have little tiny bubbles of aqueous in your organic or organic in your aqueous. And so supported liquid extraction is nice because it reduces the likelihood that an emulsion is going to form. And also, I know as most of you have done this experiment already in lab, you know that it's quite easy to do and it's pretty good for time. You're able to do it pretty quickly. And then you're then able to analyze the elutant as you would through GCMS or LCMS or whatever analytical method you're using. Some problems with supported liquid extraction is that they might not necessarily be as sensitive or as specific as something like a SPE, which we're going to talk about in a second. But in terms of speed and efficiency, supported liquid extraction has its benefits. This is what you would see in a supported liquid extraction. So pretty much you're just loading your sample that contains your analyte, your matrix, and that supported liquid extraction material. It's going to load on top of the sorbent pad, and then eventually it's going to load into it. At this point, the analyte and the SLE material is going to interact, and then eventually the analytes are going to be partitioned into the elution solvent and be collected. So all of the dirtiness that's inside of the samples is going to get stuck to the sorbent pad, and you're going to have a clean analyte um, come out with the elution solvent. So last we have our solid phase extraction. In solid phase extraction, the analytes are isolated from an aqueous sample by absorption onto a sorbent pad, followed by washing, drying, and then elution. Depending on the choice of column, the extract of the target drugs is achieved by van der Waals forces, aromatic, hydrophilic, or electrostatic interactions. Modified silica polymer and different functional groups are going to be attached to the solid phase of the column to allow for these interactions to take place. Some different types of solid phase extraction that you might encounter are reverse phase extraction, in which case the retention mechanism is a hydrophobic interaction. So the drug analyte is going to interact with the column through hydrophobic interactions. This is usually used for aqueous samples with analytes exhibiting non-polar functional groups. So again, non-polar meaning non is going to be similar to hydrophobic. So this plays on our like dissolves like or like attracts like theory that we're going to use throughout different things. So we're going to talk about it again when we talk about gas chromatography and liquid chromatography in a minute. But for right now, a lot of the stuff that we're doing in toxicology, especially in our labs, revolves around the like attracts like or like dissolves like, um, such that if we're using a hydrophobic interaction, right, we're going to be looking for a nonpolar or a hydrophobic drug. Our cations are going to work through cation exchange, and their target compounds are going to be basic drugs. Our anion exchanges are going to work through anion exchange, and these are going to be for acidic drugs. And then we'll also have mixed mode columns, which use reverse phase cation and or reverse phase and anion interact exchange molecule mechanisms. Mixed mode could be a combination of reverse phased and cation or reverse phased and anion. The drugs are going to be retained based on hydrophobic interactions and or electrostatic interactions. And this type of SPE procedure is used for the extraction of basic acidic and natural drugs. So next we're going to talk about screening. This is kind of like the first step in our process. So screening is usually going to be done through one of a couple of ways. I'm going to just talk about, I think, 3 different ways that you can do screening, but there are other screening methods available. Screening method is going to kind of give us our quick and dirty result. So a screening method is going to give us our preliminary result. And then if it's positive, it's going to later be used in a confirmation technique. Our screening methods are going to have cutoff values. So a cutoff value is going to be a certain number that is determined by the laboratory, where if it's the drug concentration is above that number in the sample, it's considered positive, and if it's lower than that cutoff value, it's considered negative. And our screening gives us this first kind of insight into this. We'll use a cutoff value also when we talk about our confirmation results, but our screening result, like I said, is going to give us that preliminary information. Is there drug in the sample? So one of the most common types of screening assays are going to be immunoassays, and this is based on the binding of a target molecule and an antigen with a corresponding antibody. Immunoassays are quick and convenient tests for many analytes in a variety of different matrices. Positive tests are then going to go on and be confirmed by a confirmation test. So you can see here pretty much the general idea behind an immunoassay. So we have our plate or our well or whatever it is going to have antibodies that are attached. There's going to be target proteins that attach to the antibodies. And then we are going to add in an enzyme labeled antibody. If there is a substrate present, right, we're going to add that in. If they are unknown, if the substrate is there, it's then going to form a complex with the radial or with the labeled enzyme, and that's then going to stick to the antibodies. And then we read the absorption of this to determine the amount, the concentration of drug in the system. So one type of an immunoassay is EMIT. EMIT stands for Enzyme Multiplied Immunoassay Technique. It is a homogeneous immunoassay, meaning that it does not require the separation of the bound and free fractions. EMIT is easily automated, but it is less sensitive than other techniques like ELISA, which we're going to talk about in a second. And the theory behind EMIT is that it is based on the enzyme activity of drug-labeled enzyme G6PDH. And this is modulated by the absorbance against the amount of drug. So the enzyme activity of the presence of glucose 6-phosphate results in the conversion of nicotinamide adenine dinucleotide, or NAD, to its reduced form, NADH. And then we're going to subsequently look at the amount of NADH at 340 nanometers through spectrophotometric analysis. The addition of the anti-drug antibody results in the binding of drug-labeled enzyme, which is then going to reduce the enzyme activity, such that if any drugs in the sample compete with the labeled drug to bind to the antibody, the unbound enzyme is going to become active and increase the absorbance at 340 nanometers. Increasing the amount of drug in the sample increases the amount of enzyme activity, and hence the increase in the rate of change of absorbance at 340 nanometers. And ELISA, that oxygen treatment therapy, sorry, shouldn't be there. Um, ELISA is enzyme-linked immunosorbent assay, and this is a heterogeneous immunoassay, meaning that it requires the separation of the bound and free antigen before the measurement of the signal. This uses a coated microplate to produce color change in the presence of certain analytes, and then the color is going to be inversely proportional to the amount of drug in the specimen. So pretty much if there is a higher amount of drug, there's going to be less intensity of the light. I know at least that the 415 students did this experiment, so they were able to see this happen firsthand. But pretty much what is going to happen in ELISA assay is you're going to take a 96 well microplate that is going to be pre-treated with the antibody. You're going to load a sample of your unknown, about 10 to 50 microliters, into the microplate, followed by a buffered solution, which is going to contain a fixed amount of drug labeled with the enzyme. The plate is going to be left for a certain amount of time to allow the enzyme labeled drug and the innate drug that was in the sample to compete for binding to the solid phase antibodies that are adhered to the well plate. After the incubation period, the microplate is going to be washed with a suitable buffer. And this is what we refer to as the separation step when we talk about the separation of the bound and free antigen. And this is going to remove all of the unbound labeled enzyme. And then we're going to add a substrate reagent to the microwell plate to develop a color. That's another incubation. After that incubation, the development of the color is stopped by the addition of a strong acid, and then the absorbance is measured. So again, this is inversely proportional, such that lighter colored wells are going to have more drug, and darker colored wells are going to have less drug. Finally, we have our lateral flow assays. The lateral flow assay is a single-use assay, and they're a variant of the ELISA assay where they run on a solid phase strip instead of inside of a well. These became really popular with COVID. So now we have a lot of COVID tests and a lot of flu tests that follow this lateral flow assay architecture. They're also commonly used in things like pregnancy tests, where this would be for the detection of a hormone instead of a pathogen or a drug. Lateral flow assays are good because they tend to develop rather quickly. They're able to run roadside or in a lab with just a few minutes, and their selectivity and specificity tends to be fairly good. So finally, we have our confirmation techniques. So for the confirmation techniques, this will be done right if you have an unknown sample that you're testing and it's positive on a screening assay. You'll go on to confirm that that drug is actually present, and you'll be able to quantify the amount of drug that's present in the sample. There's a lot of different confirmation techniques. Um, this is just going to be a very rough introduction into some of them. I know you're familiar with a lot of them from, again, having done this in lab, having done this in instrumental. So this is just going to be a very brief overview of different types of confirmation techniques that you'll see in a toxicology lab. So the first one that we're going to talk about is GC, or gas chromatography. Gas chromatography, we're going to use our um, gas as our mobile phase, and then we're going to have our stationary phase on this wound up column. You'll see here that we have a carrier gas tank and a pressure regulator. This is going to allow for the flow of the carrier gas through the GC itself. Our sample injection port is going to be where we introduce the sample. This is going to happen at a really, really, really high temperature, such that the analytes or the entire sample is going to become vaporized and then be carried by that carrier gas, which is again acting as our mobile phase through the column. There's going to be some sort of interaction with analytes in the column before it reaches the detector, which gives us our signal output. You can see here that this all is happening in a column oven, right? So this whole thing, everything is going to be at very high temperatures. The GC columns are going to be made out of steel, and they're going to contain a stationary phase, which is going to be a solid or a liquid. That's for liquid chromatography. Um, liquid, sorry, it's either going to be a solid, such as gas solid chromatography, or a liquid, such as gas liquid chromatography, which is used for partitioning. Capillary columns, which are a stationary phase with an inner coating, are going to be what we most commonly use now for our gas chromatography. And the stationary phase is going to be on fused silica. This allows us to have a high purity synthetic quartz protective coating, which is what's going to be applied to the outer surface, such that it doesn't really burn when it's in that super high oven. The volatility of the substance is the most important factor in defining its separation in gas chromatography. The more volatile the compound is, meaning the lower the boiling point, the more likely it is to be in the mobile phase, and then it's going to have a quicker retention time, so it's going to move faster out of the column. For complex mixtures with some components and varying retention characteristics, it's often impractical to just use a single column temperature, also called an isothermal run. So instead, we're going to systematically increase the column temperature throughout the analysis to dramatically reduce the amount of time that's taking higher boiling point components to elute. And this is also going to improve the sensitivity of the assay, as the peaks are going to be sharper. If analytes aren't eluting at a proper speed, the chromatography is going to be affected, and you can have peaks that are going to be more drawn out. In gas chromatography, one of the easy ways to fix this is just by increasing the temperature, right? So that things are moving through it more quickly, so that the amount of time that the sensor is detecting the presence of the drug will be kind of drawn out, will be quicker instead of being drawn out, which will make your chromatography um, not have as nice of a shape. You want to just have really sharp peaks. So there's different detectors that can be used in gas chromatography. We have a thermal conductive detector, an electron capture detector, a flame ionization detector, and a nitrogen phosphorus detector. The most common type of detector used in toxicology is going to be the flame ionization detector, or the FID. And this is used most commonly throughout toxicology because it responds to nearly all classes of organic compounds or compounds that have a carbon and hydrogen bond. Another type of detector that we use fairly often is a nitrogen phosphorus detector, and this is going to be a detector that's specialized for compounds that contain nitrogen or phosphorus. And something else to note about gas chromatography is that it requires a derivatization step. And the main reason that this is performed is because it allows us to analyze compounds not directly amenable to analysis owing to inadequate volatility or stability. So what derivatization does is it improves the analysis by improving the chromatographic behavior or the detectability of the analyte. And it does this by using salination, acetylation, or alkalization, depending on the type of drug analyte that you're looking for. Will determine which type of derivatization technique you use. So the other type of common confirmation technique that we would use is LCMS. So LC. LC refers to liquid chromatography. So we have our mobile phase, which is going to be a liquid in this case, and we're going to put it through a very high pressure pump to establish a high pressure in the system. This pressure is going to push the mobile phase through the stationary phase, which is located on a column. And you can see here that our mobile phase is going to go into the pump, and then it's going to go into the injector where it is mixed with the sample. It's then at that really, really high pressure going to be pushed through the column or the stationary phase before it reaches the detector. And then in the detector, it's going to give us our data acquisition. You can see here that this directly goes into a liquid waste. So after the liquid has been pumped throughout the instrument, it's then going to go into a waste bottle. So if you are running LC analysis, this is something that you just have to be aware of. The chromatographic principles for LC analysis are retention time, capacity factor, and retention time relative to internal standard, as well as column selectivity and the resolution. So there's many different types of liquid chromatography, with the most common being polarity-based chromatography. And again, this pulls in our like attracts like theory. So in polarity-based chromatography, if the stationary phase is if the stationary phase is polar, it's going to be called normal phase chromatography. And in this case, polar compounds are going to be more attracted to the stationary phase. So they're going to have longer retention times because they're going to be interacting more with that stationary phase than they are with the mobile phase. Usually, if you're using a stationary mobile phase that is, sorry, a stationary phase that is polar, you're going to be using a non-polar mobile phase. On the other hand, if the stationary phase is non-polar, it's going to be called reverse phase chromatography. And then again, in this case, the non-polar compounds are going to have a higher likelihood to stay in the stationary phase. The mobile phase for normal phase chromatography, or which is when it's polar, is sometimes also going to be polar, in which case it's going to just kind of change the interaction to see which the analytes are going to want to stick with more, either the polarity of the mobile phase or of the stationary phase. Um, Sorry, I wrote that wrong. In our normal phase chromatography, the stationary phase is polar, or silica, and the mobile phase is nonpolar. The absorption chromatography, separation is controlled by the competition between solute and molecules, or molecules and the mobile phase for the absorption sites on the silica surface. Polar groups are going to be most strongly attracted to these sites. So then polar compounds are going to be retained more strongly than nonpolar ones. In reverse phased chromatography for the mobile phase, in reverse phase chromatography, the stationary phase is going to be nonpolar, and then the mobile phase is going to be polar. In this case, retention is controlled by the hydrophobic interactions between the drugs and the groups on the stationary phase packing material. The retention is going to increase as the analyte decreases in polarity. So again, sorry, I definitely confused you guys with the normal phase. So normal phase, the stationary phase is polar and the mobile phase is nonpolar. Separation is controlled by the competition between solute molecules and molecules in the mobile phase for the absorption sites on the surface of the column. Polar groups are going to be attracted most strongly to these sites and therefore be retained longer than nonpolar ones. And then the reverse phase is going to be the opposite. So, just a detector that I wanted to talk about quickly that occurs most commonly when we're doing LCM or LC or GC is the mass spectrometry. So, mass spectrometry is the analytical technique that measures the mass to charge ratio of ions, and we use this to identify, quantify, and determine the structure of molecules. This process involves ionizing a sample, separating the resulting ions by their mass to charge ratio in a mass analyzer, and then detecting them. Sometimes we'll do this we use tandem mass spectrometry, which is LCMSMS or GCMSMS. And the most common type of tandem mass spectrometry is going to be the triple quad analyzer. In this, the quadrupoles are going to be mass analyzers that are formed by four rods arranged in a parallel and opposite arrangement. This creates two varying electrostatic fields, one with a direct current and one at a varying radio frequency. And these are going to be applied at right angles to each other, such that the four rods kind of create that circle. This creates a resonance frequency for each mass to charge each mass to charge value in a mass spectrum. Between the analyzers, there is a collision cell, and in the collision cell, collision energy and gas molecule fragments are going to make the molecules break apart into a precursor ion and a product ion. The concentration or the quantity of these ions is then going to be measured and then compared against each other to quantify the results. So that was carbon monoxide and then a brief introduction into analytical. If you guys have any questions, you're welcome to email me. My email is allewis@johnj.cuny.edu. Or, of course, ask Dr. Strip if you have any other questions. But I hope you guys have a great rest of your day. Thanks for watching.